The MATADOR trial was designed back in 2004. And it was a trial where we combined dose-dense AC 6 times versus 6 times Docetaxel and AC. So the only difference was a taxane, yes or no. The other difference was that in the 6 times dose-dense AC, it was once every 2 weeks, and in TAC it was once every 3 weeks. At the time, we were really interested to find patients who would really derive benefit from a taxane, because we know from the Oxford overview that in fact only an absolute 3% extra patients is cured at 10 years by adding a taxane to the regimen. Well, the issue with taxanes is that around 50% of patients develops polyneuropathy, and in many patients that does not go away. So it's really a cumbersome toxicity. So we thought if we can develop a test that can really tease out the patients who benefit, that would be useful. And so we asked all patients, all HER2-negative patients, to participate in this early breast cancer trial. And so they received either one or the other adjuvant chemotherapy regimen. And then we recollected all the tumors to generate gene expression profiles on all these tumors. And we hoped that, in fact, by doing this, we would tease out a certain profile that could predict the benefit of taxanes. Now, by collaborating with a very nice colleague in my lab, his name is Jacob van Renen, who had just developed a compendium of gene expression profiles based on single cells derived from breast tumors. He had a catalog of what is the contribution of each cell type in the tumor to a bulk RNA profile. And so by asking the computer, okay, now you have this bulk RNA profile, it comes from all these different cell types within a tumor. Can you tell me what is the composition of this breast tumor? And then the computer can tell, well, there are so many tumor cells, there are this and this cell type, so triple negative or luminal. There are so many immune cells, and there are also connective tissue cells. And so we could really stratify the different breast tumors into different, they had different compositions. And then we asked, is there any difference between what kind of chemo the patient has received and their outcome if we look at the composition of the tumors? And then we found, in fact, that only those tumors where there were a lot of immune cells present, that those tumors and patients with those tumors did very well on a taxane-containing regimen. Well, if there were not many immune cells, then they did equally well on dose-dense AC or a Docetaxel-containing regimen. So, okay, this is interesting, but we don't know whether it's true. And is there a simpler way to know whether there are immune cells in the tumor? And so then we went to the pathologist, and he scored all the tumors with the microscope and looked at the TILs, tumor-infiltrating lymphocytes. And in fact, there was a much simpler method to also find out that tumors with high TILs, in this case 20% or more TILs, derive substantial benefit from taxanes, while the others don't. And then Jacob van Renen was really intrigued, why is this? Because it's really strange that what is the relationship between immune cells and taxanes? Because we had always learned that taxanes work because they cause mitotic arrest. They cause polymerization of the tubulin, and that's their working mechanism. So what have immune cells to do with this? So what he did is he took a mouse model, a breast cancer mouse model, and he took out all the T cells from that mouse, and then he looked whether a taxane could induce a response to the breast tumor in that mouse, and it couldn't. So he thought, okay, it's the T cells, so the taxanes are doing something to the T cells. So the next thing he did was he took out the T cells from a mouse, treated these cells with docetaxel outside the mouse and then reinfused the T cells. And that induced a response to the tumor in the mouse. And then the next step was to also expose the T cells outside the mouse to the docetaxel. And then we only take the supernatant from the T cells and reintroduce the supernatant into the mouse. And that induced the response as well. So in fact, what we now know, based on those experiments, is that taxanes not only work by causing mitotic arrest, as we already knew, but they also work by stimulating the immune system to eradicate the tumor cells.